welcome to the second short course session on computational tools for genome design and analytics. The topic for today is a focus of the Paulson Lab for the last, I want to say, 4 or 5 years now, which is utilizing independent component analysis to analyze gene expression data. It's been a particularly successful way to understand what gene expression data is telling us about the cellular state. And the topic or the organization for today is roughly broken down into the first part, having a focus on how do we actually calculate ICA for a gene expression compendium. So step by step, like why do we want to use ICA to analyze gene expression data, and then what are the advantages of ICA over other methods, and why does it work so well, and finally culminating in sort of a step-by-step guide to actually calculating ICA. And as an exercise for this part, we'll be going through the Modulome GitHub repository, which has also a step-by-step guide for going all the way from downloading any available RNA-seq data for a particular organism, annotating it, calculating imodulons, and then annotating those imodulons, which are the ICA components. And then in the second part, we'll be going into more of ICA analysis. So how do we actually understand what ICA is telling us about our data set, and what does it tell us about transcription regulation in our organism as a whole? So in other words, what can we learn from this analysis? And so I'll go through some general principles of how do we use ICA for analysis, as well as different visualization methods that help that process. And then I have quite a long list of case studies, and we'll see how many of these we get through in the interest of time. I think we could probably have a shorter session today, which would be nice. I know 4 hours is a long time for anything, sitting down and working on a single topic. So I'll try to keep things a little bit shorter to the extent possible. But before I move on, I want to give credit where credit is due. There's an amazing ICA team in this lab that was started by Anand Sastry, who first developed this approach for gene expression analysis as a graduate student, and then built this whole infrastructure from the ground up. And then Sagat, Kevin, and Cam have sort of followed in his footsteps and built really nice software tools for doing this. And I'll be showcasing essentially all of their work here today. Okay, so just a backdrop for what the actual challenge here is. So the transcription regulatory network of a bacteria like E. coli is the main method that it can actually control its physiological state. Your cell can't directly change any metabolite levels, directly change DNA levels, so on and so forth. All it can really do through the central dogma is change protein levels. And it does so by affecting their expression in various ways. The focus of this is really on this type of analysis of the transcription regulatory network is really on transcription initiation. There are a variety of other methods that of gene expression regulation that sometimes surface, but you can think of transcription initiation as the predominant bottleneck in gene expression. So somehow you could see that a typical transcription regulatory network is quite complex, and these transcription factors that regulate different genes sense different signals and also co-regulate each other. So it's a hierarchical network. And this has really been a tough nut to crack for systems biology. There's been a long-standing interest in understanding how to model this network, how to really quantitatively understand it. But due to the complexity, there's 4,000 genes. Even in a simple organism like E. coli, there's 300 transcription factors, but only about half of them have been characterized. There's 7 different sigma factors that recruit the polymerase to different core promoter elements. And maybe more importantly, it's not clear to what extent every transcription regulator is actually going to be regulating each gene. It's not like a Boolean network that just flips things on and off. A lot of times these are quantitative and subtle regulatory changes that just balance the cell and keep things in steady state as it senses different kinds of pressure in its environment. So quantitatively, this has been quite difficult to understand the function of this network. And this is another way to think about this problem. So one of the main data sets that really has been flourishing in the wake of the sequencing revolution, we have more and more sequencing data, is RNA sequencing, which gives us an idea of the gene expression at any given state in our cell. And this is a big improvement over the microarray data that we used to use for gene expression data. There's less bias, you have finer resolution. And so we have really clean gene expression profiles. But the gene expression is, for any given gene, is the combination of all the activities of the different transcription factors, as well as some global factors. And these are taking in many different inputs, as I just mentioned, that affect their activity. So it still really has been a black box to understand how the measured gene expression, which we have more and more clean data, actually quantitatively is a result of the whatever computation that the transcription regulatory network is doing to understand the signals that the cell is actually sensing. So the challenge that motivated this whole analysis, this ICA analysis of gene expression data, is kind of, can we go in reverse? Can we take our gene expression data and work backwards to figure out what the state of our transcription regulatory network is, and then maybe figure out how it's integrating different signals? So another way of thinking about this is that, just kind of describing what I was saying, is that we have transcription factors that some are constitutive, like sigma factors. Some require an activating metabolite, like iron or central metabolites, in order to convert the transcription factor to an active form. And then it'll bind to your gene and affect expression, either positively or negatively, right? There's some function of the states here, whether it's bound or unbound to the gene, whether it's bound or unbound to its activating metabolite, that results in an average level of transcription for that particular gene. And then the sum of this, of all transcription factors in front of a gene, results in your gene expression that you measure at a system-wide level. So multiple regulators affecting the gene potentially under a different state, and you just get some kind of population average as your data. And this is then, regulating your, these gene expression changes regulate your metabolic network and other networks, and then changes here then have some kind of feedback here. And so it's not even a one-way thing, it's a hierarchical structure. And these transcription factors are also themselves regulated, and you can annotate their binding sites with chromatin immunoprecipitation experiments and identify where they're binding. But again, going from where they're binding to quantitatively, how much are they affecting expression in one state versus the other, is just a very difficult problem. And it's not straightforward at all to build models that fully encapsulate all of these processes. So what people have traditionally done then with all of this RNA-seq data is some maybe simpler methods. What is available without that kind of deeper understanding of all these interconnections, which is largely comes down to statistical analyses. So you may be familiar with the volcano plot, which is shown in the upper right for a differential gene expression analysis. You have on the x-axis your log fold change, so how much is the gene changing? And you have a log odd score for how significant it is, and the things in the extremes are what you would call a differentially expressed gene. And on a gene by gene basis, this is perfectly fine. You could say these genes are going up, these genes are going down, and that is certainly a partial answer to what is going on in the cell. But it doesn't really answer that more fundamental question of what regulators are active in the cell, or what signals are the cell sensing in terms of stresses or nutrient availabilities, things like that. And if it's really not a very simple regulatory situation where you have one gene, one regulator, one signal, which often it is not that case, it's really hard to go from this kind of plot to that deeper understanding. A bit closer to that deeper understanding are methods that try to use network analysis or pathway analysis to integrate multiple gene changes that affect some common function. So oftentimes here you're doing something like encoding your known transcription regulatory network and saying, are there changes enriched in these regulons? Or there's even more formal network component analysis that actually tries to perform a decomposition using the known network. But that really is this kind of approach, while more powerful and closer to that goal of fundamental understanding, really relies on your prior knowledge. And so if there's any kind of incompleteness with our prior knowledge, I just mentioned that only 150 out of 300 transcription factors are even characterized. So if you're missing that knowledge, then these kinds of methods are really not going to be the best tools for discovery, for understanding new regulation. And then another method or family of methods are different unsupervised machine learning methods, which brings us to the topic that I'll be talking about today. And at the simplest level, you might be familiar with clustering. You can always perform, for example, hierarchical clustering on a gene expression data set and see if there are particular genes that group together and move up and down together, have some kind of correlated function. That can often be informative, or particular conditions that have very similar profiles, and that can also be informative. And we do use these kinds of methods in combination with basically everything else. Also very popular these days are low dimensional visualization methods like TSNE and UMAP. These are, they inherently perform some clustering while also simplifying the representation of the data from thousands of variables down to just 2 dimensions that maybe your conditions cluster by that, and gives you kind of a snapshot of the major drivers. But it doesn't, you're in 2 or 3 dimensions in these cases, as often is the case, and you still have the activity of 300 transcription factors contributing to that, right? So it's not really a fundamental breakdown inherently. You still have to go further beyond what these can do in order to get that kind of input-output understanding that really is the goal of this transcription regulatory network analysis. And then the last family of methods, and the one that's I'll be talking about the most today, is matrix decomposition. And this is when we're taking our RNA-seq data set and breaking it into separate matrices that have an interpretation that actually is very close to where we're getting at. And specifically, I'll be talking about the most successful method that we found for doing this. And we've tried all of the methods that we're aware of, and that's independent component analysis. So what is matrix decomposition? Why do we want to do this? So we have our gene expression data set. Our rows are genes and our columns are samples. So this is not just a single gene expression data set, it's a compendium of data. And what we're hoping to do is infer the transcription regulatory network structure and activities by breaking this matrix into 2 separate matrices that represent those 2 components. So the first matrix is a structural matrix. It's going to contain the structure of the transcription regulatory network if this decomposition works. And the second matrix contains the activities in a sample condition specific manner. of each of these components. So there's a one-to-one correspondence between rows and columns of these two matrices. But when you multiply them together, they should be able to reproduce your original data set. So if you can find a decomposition that actually does this, that where your one matrix has structure and the other has activities, this actually does address that, that original goal, which is understanding how different regulatory activities are combining to result in your expression. This just explicitly does that. But the question is, can we find a decomposition that actually does that? Right? This, you can break a matrix down a lot of different ways into separate matrices, and they're not all equally successful. And so there's several different matrix decompositions that have been studied extensively. Probably the most well-known is principal component analysis. And each of these methods has a different constraints and criteria that they use in order to solve this problem, to break one matrix into two matrices. Now for PCA, the objective of the method is to maximize how much variance is explained by your components. So your first component is going to explain the most variance possible. It's going to be the thing that is changing most in your expression data set will be captured in your first component. Your second component will be the second most, subject to the constraint that it is all correlated with your first component. So it's kind of a highly constrained version of matrix decomposition. ICA has a slightly different objective. And instead of trying to maximize how much variance is explained by the top component, it just tries to maximize the statistical independence of any components that you have. So it tries to minimize any kind of relationship between your components in a statistical mutual information sense. And then last, non-negative matrix factorization tries to come up with a breakdown such that both of these matrices are non-negative. So there's no negative values in either of these matrices. Why would you want to do that? Well, this is often used in image representation. Anytime that you have a parts-based representation of your data, non-negative matrix factorization is a very intuitive way of doing that. So those are the three most commonly used methods, but let's talk a little bit about ICA in particular, and then I'll contrast it with PCA to describe or intuitively try to justify why this is such a successful method. So ICA is motivated by the problem of blind source separation. This is kind of the commonly described story here is a cocktail party where you have multiple different people talking, and you have microphones placed at different parts of the room. So you don't have a microphone in front of each person. They're at different locations than the speakers. And so each microphone is picking up a mix of all of the different speakers, some more, some less, depending on how close they are. So you have a mixed kind of measurement here. And what ICA tries to do is break this back down into the original signals to figure out what everyone is saying. So the signal is the source, and then the activity is the volume at which they're speaking. So that's the intuition. By biological analogy, the speakers in this case are transcription factors, right? So they have different levels of activity, which is how loud they're speaking on a particular condition. And all of these will then combine if they regulate a gene. If you have multiple transcription factors regulating a gene, they'll combine their signals to result in the gene expression or the differential gene expression relative to some reference point. So that's how this is. ICA is motivated intuitively. And let's go into a little bit more about what these matrices actually contain. So as I mentioned, this is a matrix decomposition of our original gene expression data, which is genes by conditions or by samples. And we're breaking it down into two matrices. And I apologize for the notation. In classical ICA literature, these matrices are S and A for source and activities, or signals and activities. But we've started using M for this matrix. So you might see M here because we call these I-modulons for independently modulated sets of genes. Anyways, that nomenclature aside, let's go into what is in these matrices. So first, our structural matrix, which is either S or M, contains our regulator signals. In other words, so it's genes by signals or components. And each gene here you can think of as a regulon. It's a set of genes that move together. And so the intuition is that they must have some common component that is actually regulating their activity, such as a transcription factor, such as a shared transcription factor. And then the second matrix, the activity matrix, then is signals by conditions. So it reproduces matrix multiplication genes by conditions when you multiply them together. And this is how active each component is on particular conditions. And so the intuitive expectation is that when this transcription factor is active, it would have a particularly high activity. And when it's inactive, it would have a negligible activity. So that's the expectation. If this all works, and I'll show case studies later for evidence that it does indeed work well. So just an example of what this actually looks like. So when we calculate this on a real data set, we get a lot of genes that have near zero values. And we can filter these out and say they're effectively not in that component. But any gene that passes a threshold can be is classified as part of this component. And as a preview, you can see in this particular case, a variety of GLP genes all grouped together, which is some indication that this is a biologically meaningful component. On the activity side, you can look at the activity of this component, the GLP component that I just highlighted, and you can see its activity on most conditions versus its activity on glycerol, particularly in particular conditions. And this is a glycerol regulator, and as you would expect, the activity is very high. So just as a quick preview of the deeper results later, you can already see that this biological intuition, that intuitive breakdown that we are looking for of TRN structure with TRN activity combining to result in gene expression is actually panning out. Okay, so let's go into a little bit on ICA versus PCA. Why does ICA work so well, and why have we not had success with other methods? With PCA as an example. So principal component analysis, as I mentioned, identifies orthogonal directions of maximal variance. What does that mean? Orthogonal means that they have to be at right angles to each other, and maximal variance means that the first component, in this particular case, means that the first component will be pointing in the direction of the most variance in your data set, whatever direction is changing the most. And then subject to this orthogonality constraint, your second component will point in the second most variable direction. So you can see here, if we have some true independent data source, our direction of maximal variance might be something like here. And then subject to orthogonality, sorry, you can see it here. So in the upper right, we have our observations. So we have the true independent sources, but then we have our mixed sources, which is a combination of the two with some variation that actually puts it off the axes. Now PCA, when it's trying to understand these directions of variance, will first point in the direction of maximal variance, which is this direction here that has the most change to it. Now, when it's looking for a second component, because there's only two components here, it's in 2 dimensions, it has to be at a right angle to it. So in this case, it's going to be pointing in this direction no matter what. And you can see it's kind of non-ideal because it didn't capture this, that the fact that the second direction of variance is not actually at a right angle to the first. So that orthogonality constraint is actually detrimental to understanding how these signals are actually combining. ICA, though, doesn't require this direction of maximal variance. It just requires statistical independence. So what does statistical independence mean? That's actually shown on the bottom left. So we're all familiar with what correlation is. So correlation is one way that signals can be dependent. So if you know the idea behind mutual information or dependence is that if you know something about one variable, it tells you something about your other variable. So in this case, you know if your x variable is low, your y variable is also low. And you know if your x variable is high, your y variable is also high. This is correlated and it's also dependent. But it's also possible to be uncorrelated and still be dependent. So in this case, if you know that your x variable is low, your y variable is high. But if your x variable is high, your y variable is also high, right? So it's still, your variables tell you something about each other, but it doesn't have to be a simple linear correlation. So statistical dependence is a more powerful way of describing relationships between data. There is no way to be correlated and independent, so that's why there's an X here, but you can see the uncorrelated independent case here. So all ICA requires is that your signals that you're looking for, the components you're trying to break down, should be uncorrelated with each other. So they should not share information. In other words, they shouldn't share genes, is another way of thinking about it. So when ICA looks for dimensions, it actually finds that there are two main directions of variance, and it doesn't require a right angle between them. So ICA can actually recover the proper true independent signals, whereas PCA, because of this requirement, has kind of this slant to it. So that's an intuitive understanding for why ICA is more flexible than PCA and actually ends up recovering these linear combinations of signals in a more meaningful way than PCA does, because PCA, through this orthogonality constraint, is just over-constraining in how it breaks down a system. Not to say that PCA—
Can I ask a question?
Yes, please.
Sorry, I don't want to interrupt, but you said the components should not have the same genes in them, right?
Yes.
Does that make it also a bit of a handicap because there might be multiple transcription factors acting on the same gene?
Yeah, and this breakdown isn't actually perfect at the end of the day. So, so as you'll see, there are these ICA components that are correlated with each other and they cluster together. And a lot of times that has to do with like a master regulator or a stimulon, some kind of shared function. There's all kinds of complexities that don't break down linearly, and that's kind of I would actually say it is a flaw of ICA still, is that we're still linearly combining signals, and in reality, biology has all kinds of nonlinear things that we're not fully capturing.
Yeah, thank you.
Okay, so if you now believe me that ICA is a good method for doing this kind of matrix breakdown, let's talk a little bit about how it actually works. So the fast ICA algorithm is the method that we still use. Sorry, I just want to make sure to click through everything. So first, let's describe what we're seeing here. So this is a signal mixing. So we have two original signals that are the true things that we're trying to capture. In this case, these are, not a particularly biological example, but it's just a mathematical example to demonstrate how this algorithm works. So what we'd like to do is to understand that these are the two sources of our signals. However, they actually got combined, so they've been mixed with different volumes, different strengths. to result in a mixed signal. So one thing that you notice, and this is a central limit theorem thing, and this is one of the keys for why IC actually works, is that mixed signals actually tend to look Gaussian. So your original signals are highly non-Gaussian, right? This is like anti-Gaussian, this is like uniform. But when you mix signals, they actually tend to look more Gaussian. And that's the numerical trick that we actually use to solve this matrix decomposition. So first, you can always assume you're starting with your mixed data, right? That's what our gene expression data is. It's a mix of the activities of everything. So we center it on 0, and then we whiten the data. So we remove any existing correlations using SVD. What does this do? So going from mixed to whitened, you can see it looks like it goes from here to here. So what it's done is kind of equalize the magnitudes of the different signals. Numerically, this simplifies the search problem because now all the ICA finding algorithm has to do is rotate this. So if you remove the strength of these signals, you then just have to rotate it. So you can see it's kind of stretched these out in a way that now it matches the signal better. And then finally, you just rotate. And how do you know when to stop rotating? How do you know how to actually rotate this? Well, you rotate it in a way that minimizes how Gaussian the mixed distribution actually looks, based on this principle that any mixed signal should look Gaussian. So if you minimize how Gaussian it looks, you should be separating the signals. You should be unmixing the signals, okay? So that's all there is to this algorithm. To go from mixed to original, you first whiten your data, which makes it, simplifies the problem. So all you have to do is rotate it. And then you rotate it in a way that minimizes how Gaussian your signals are. And you can see in this particular case that actually recovers your a pretty good representation of your original signals. So that's how ICA works. We don't have to go too much into this, but there are different metrics to actually measure how non-Gaussian something is. So there are peaks that are considered too sharp or too unsharp, called the kurtosis of different distributions that you can measure. So a Gaussian distribution has a kurtosis of 0, so you can try to minimize or maximize that. That's not particularly robust though. There's this term that's called negentropy that is actually more commonly used because a Gaussian distribution, so entropy is an idea of the randomness, right? And the Gaussian distribution has maximum entropy. It's, as I just mentioned, if you mix signals together, that's adding entropy, and that increases your entropy and it makes it more Gaussian. So it makes sense that a Gaussian distribution has a lot of entropy. And so you have an approximating function then. So you can't directly calculate negentropy, but you can calculate an approximating function, and this is actually what we end up using. And so this is our objective criteria for the rotation, is this negentropy estimate. Okay, so that's the ICA algorithm in a nutshell. What's wrong with it, right? What can't it do? Are there any limitations? So as Emre already mentioned, the requirement that there's minimal overlap is not necessarily always correct. So the most clear way of saying this is that because you're minimizing Gaussian sources, you're always going to force things to be as uncorrelated and separate as possible. But in reality, you might have situations where there is overlap in regulons due to whatever factors. And so this will kind of hide those, or it'll actually, what it'll tend to do is it'll separate a single global regulator into like linearly separate components. So it'll break down these nonlinear effects or non-independent effects into separate independent effects, which you may or may not want. It kind of complicates the analysis because then you have more components to figure out. Another key fact that I didn't mention so far is that ICA is stochastic. So PCA, you can calculate it definitively and never have any kind of question mark really about whether this is the real principal component analysis breakdown of your matrix. ICA is not like that. So it derives different approximations of the your globally optimal solution. But it is inherently stochastic. And so in practice, we have to run this tens of times. And what we do is we keep the components that we see over and over across different runs. So there's a clustering step at the end of this sampling procedure that I'll describe later. Also contrasting with PCA. So as I mentioned with the description of PCA, your first component is going to have the maximum amount of variance. ICA doesn't give you any kind of order or direction to your components. It doesn't tell you what's positive or negative either. So you have to do your own ordering. So you have to, for example, calculate how much of your original variance is explained by each component, and maybe then have some kind of cutoffs on what you keep, as well as these clustering methods, alongside those clustering methods, to figure out which ICA components are most significant. And in that same token, it doesn't inherently reduce the dimensionality. So PCA is often used for dimensionality reduction for numerical purposes, or just to understand your system in fewer dimensions. ICA doesn't do that. It's going to give you a full set of components for your data, to reproduce your data, and it's up to you to figure out how many of those components are actually meaningful and how many are noise. Okay, so on that note of explained variance, what is that talking about? How do we tell how much of our, if we do this this clustering method, how do we, or if we do some kind of filtering method and we don't have a full breakdown of our matrix, how can we tell how much of our original data matrix is reproduced by a particular ICA breakdown? Well, we can just compare the original with the recombined ICA breakdown. And oftentimes, so you can see as you add components, the fraction of your original data that you're explaining goes up and up and up, but it kind of plateaus. And so we can use this kind of plot to sometimes set a cutoff on how many components we actually want to take. And also we can, but this inherently will order the components, so we can identify which components are changing the most or contributing the most to differential expression in our particular data set. Okay, so now let's talk a little bit more about that, that clustering step. So ICA is stochastic. Every time we run it, we're going to get a slightly different answer. It's pretty discouraging if you have a story around a particular ICA component, you think it's meaningful, and then all of a sudden it disappears when you run it again. So how do we get robust components? How do we make sure that our answer of what the TRN structure is actually stable? So first we start off by running ICA a certain number of times. Around 100 has been successful in practice for E. coli. This might change depending on your organism, your data set, so on and so forth. And then you can cluster your data. So you can find the distance between the components across multiple runs. So you can find what is the closest component to this in this other run. And you actually can cluster those and then maybe find the centroid of the different clusters to find what your real estimate of the component is. But what about things that don't cluster very well? Do we still call them components? How do we know what the cutoff is? Well, there are algorithms like dbScan that are density-based clustering. And what a density-based clustering algorithm can do, what density means is that if it sees a lot of points with neighbors, so these points that are red are in the core of the algorithm because there are other red points that are also within a particular radius, right? So you can see this is dense, and these circles are that radius that it's looking for other core points around it. Now, if your a peripheral point, and maybe you only have one core point within your radius, you get classified as a border point. And if you have no points, no other points in your radius, you're classified as noise. And so this kind of radius-based clustering called density-based clustering, dbSCAN is a very common method for this, inherently allows us to filter out these points that maybe only show up in 1 or 2 runs. Maybe they have a high amount of variance across, there's no near neighbors to it. And then we can just filter those out. And so those are unstable components, and we just remove them from any analysis, and we don't consider them as part of the final independent component set. So that's kind of the last piece. This is just an example of the FastICA breakdown in scikit-learn, which is the Python machine learning package. You don't have to tell it the number of components. Obviously, you're going to want to run it in parallel. You can see there's a white and true here. This log cosh is that neg entropy function, number of iterations, tolerance, so on and so forth. So this is typically the set of variables, and these are the same issues that I described previously. So you can see they're all kind of mentioned here in this function call. So once we have our ICA breakdown, as I mentioned, we have these, it's a continuous matrix, right? It's not just binary ones and zeros, but a TRN structure really should be ones and zeros. Can you be half in a regulon, half regulated by a gene? Well, you might be stronger regulated, right? But you should be able to definitively say this gene is not regulated by a transcription factor, right? So how do we identify these thresholds when we're defining a regulon based on an ICA breakdown? Well, it comes down to some kind of quantitative cutoff. So I showed a plot earlier in your activity matrix. Oftentimes, if you take a histogram of the number of genes versus their coefficient in a particular modulon or ICA component, there's going to be a lot of near zero values. So you're going to have this peak around 0. And then you're going to have some values that are higher. And the question is, at what point along the x-axis do you start saying, okay, this is regulated, and this is in this regulon? And so it comes down to figuring out your threshold. And so there's 2 methods that, that you might think of. The more sophisticated one is called the D'Agostino method. And what you do is you try to, you assume that your base distribution is normally distributed. So whatever null distribution you have, or whatever real distribution, is a combination of a null distribution and then some actual signal. So if you remove the highest points, the extreme points on the left and right, you, at a certain point, you should be left with a normal distribution again, right? So that's what this method is. You remove the highly weighted genes until what remains is normally distributed. And so this requires a cutoff, but we've actually optimized this cutoff in practice by figuring out cutoffs at which the genes you've removed, which are the ones in the regulon, have a really high degree of overlap with your TRN. So kind of phenomenologically looking at what works in predicting regulons, we've been able to find a cutoff that, that works for this. Another more naive way is to just assume that you have different clusters. So you have one cluster around 0, and then you have different extreme clusters. And the benefit of this is that it's non-parametric in that you don't really need a cutoff, you don't really need a parameter or a cutoff other than how many clusters you want. But in practice, it doesn't quite work as well. So that's kind of the last piece of what it takes to actually calculate your ICA components. So just to summarize this workflow, you start with an RNA-seq compendium, and you perform this ICA breakdown with FastICA, running it hundreds of times in clustering, and you end up with 2 matrices. Your S or M matrix is where your regulators are regulating. So your regulon structures are in your components. And then your regulator activity levels are in your A matrix. And you can apply a threshold to say, yes, this is in this regulon or not. And then when you're left with that, now you can proceed to all the downstream analyses of what these regulons mean. And that's the topic for the second part of this workshop. So I won't go into this too much, but just to preview it, you can map these components, which we call imodulons now, because they're independently modulated sets of genes. You can map them to the known TRN and say, is there an enrichment of these imodulon genes with the known regulon, which would say, okay, this is probably regulated by the same transcription factor there. You can actually go in and validate whether these predicted genes have a binding site for a transcription factor in front of them with the ChIP-EXO experiment. And then you can validate the regulator activity by seeing if the activity of this imodulon changes in a way that's expected as you go to conditions that are supposed to be activating this transcription factor. So if you have an iron starvation transcription factor, and you put your cell in an iron starvation condition, you should see this imodulon light up. If not, then you probably don't have the right interpretation of that imodulon. Okay, so that's it for the description of how these imodulons are calculated. For the last part of this, I just wanted to talk a little bit about the data that goes into this process. So originally, I said you start with this RNA compendium, RNA expression compendium. Where do you get that from? Well, there's quite a lot of data that is publicly available now, NCBI-GEO and the NCBI-SRA. And so our lab has been annotating how much of this data is available for different organisms, and we've been building RNA-seq and microarray compendiums for these different organisms to perform ICA. But just a little bit on data quality, not all data is equal. It's well known that different labs have different, can contribute different batch effects to data sets, and all of this can create issues. So our lab has developed for E. coli a database called PRECISE, which is an in-house set of high quality RNA-seq data. Compared to all public data sets in GEO, we're now nearing 1,000 in-house data sets, which you can see is quite a substantial fraction of all available data. I don't have the latest 2021 numbers here. But the fact that we have a single data set that is all using the same protocols for in the lab as well as post-processing has resulted in an RNA compendium that has a median R squared for replicates of 0.98, which is extremely high fidelity. So this type of data is extremely valuable for performing an RNA-seq breakdown, because then you're breaking down real signal and not batch effects and things like that. So you can see here the replicates versus different conditions. The replicates are clearly quite separate. So let's just talk a little bit about as you scale up your data, what happens. So as you add expression profiles, the number of imodulons you actually calculate increases as well. But interestingly, at a certain point, the number of regulatory imodulons, meaning the number of imodulons that you can associate with a regulator by enrichment with the known regulons of those transcription factors, kind of plateaus. What this would suggest is that we're kind of arriving at a stable representation of what our TRN actually is. Just describing what this actually looks like, and you can see these diminishing returns here. These genomic imodulons are due to things like just deletions of genes that kind of cause these non-physiological transcriptional responses. And also, interestingly, this number of uncharacterized imodulons is kind of flat, which could suggest that either there's a finite number of unknown regulators, or this is kind of some basal noise that is getting added in here. This is just another way of looking at the same thing. So if we go from 124 profiles to 168, 198, these are the imodulons that you get, and you can see these are the ones that continue through. This one, for example, splits into two, so you can get interesting things like that. Then you have other imodulons that only show up at higher expression levels, but you can see that the number that you're adding kind of plateaus. So that's, but I guess the positive thing about this type of plot is that the majority of your imodulons are remaining stable even as you add data sets. So you're not just adding a set of 20 more conditions and calculating a completely different TRN. It's actually fairly stable. And so, yeah, so with this type of analysis, then you can even identify certain new conditions that activate a that are associated, for example, with a knockout of an unknown gene. This is maybe closer to what you can actually do with ICA. But with this type of data set, you can actually annotate new genes, because you could say, okay, a knockout of this gene resulted in this uptick of particular activities. And that can give you some idea of what this gene is actually doing physiologically. And this resulted in the annotation of 3 poorly characterized transcription factors and 3 completely uncharacterized transcription factors with this type of workflow. Just one last note on adding data. So normally when we add data to this precise data set, we'll just recalculate the imodulons and go through this whole procedure. But it is quite time consuming, and there is some annotation involved with making sure that we understand what all the imodulons actually are, how to interpret them. So sometimes we don't want to go through that whole procedure. We don't want to recalculate our ICA, but we do have a new data set. We want to understand what the activities are of these different ICA components that we do know about. And so we can actually use our existing breakdown of ICA to analyze a new data set. And we do that with a simple matrix operation here, taking an inverse. So if we have our X prime, that's and combination of, so this is our new data. So we use our original N matrix from ICA, but we're trying to infer what the activity is on this particular condition. So it's an A prime here. And if we just take the inverse of this, we can get an estimate of this A prime. So matrix inverses are not the numerically best approach in the world, but assuming you can get a stable approximation of this, you can infer what your activities are on a new condition without having to recalculate everything. So this is a good procedure to know about. Just one last component on data quality. So why is it so important that we have high fidelity data? Well, if you have low fidelity data, some replicates in the off diagonal, so on and so forth, these can actually end up as their own imodulon. And so it's not a meaningful imodulon, it's an imodulon that has some kind of experimental noise in it, some kind of batch effect. And you can subtract that out after the fact if you want to do that, but it's not a biologically meaningful signal. And these kinds of things can also result in inconsistent mapping. Here you have this one cluster, but you have this outlier here showing up. Okay, so that brings me to the end of this lecture component right on time. Just to get into a little bit of the overview of the exercise that I'll be walking through. First, I want to go through imodulon DB, which is Kevin Michel's wonderful website for viewing imodulons, just to get a more intuitive understanding of what these variables are, get them in your own hands. So I'll just walk through that, and hopefully all of you can walk through and click through this website as well. And then secondly, because we're talking about computation here, I want to step through the Modulome workflow, which is hosted at Anand's GitHub currently. If anyone doesn't have this, let me put this in the chat in case anyone is not already set up with this. You could at least follow along on the website, but I would encourage you to clone this repository and try to work with these notebooks in on your own. That said, there are components of this that are require more extensive installations of Docker and a Nextflow workflow for RNA-seq analysis. So I wouldn't expect that you're going to do all of this, certainly not live, but you might not even want to do this because this goes through the full setup all the way from downloading data through RNA-seq analysis and then ICA calculation. So, so I'll also point you at a couple particular workflows that are relevant if you already have a data set, and those are easier to execute without a server, without waiting a long time. Okay, any questions before I move on to the exercise? Okay, if not, let's go ahead and take a look at what iModulonDB is. So iModulonDB is a website that Kevin Rachel developed for, sorry, putting this in the chat, for viewing iModulons. So it's intended to increase the accessibility of this. It has a really extensive about page. So if anything I'm saying is unclear or you want to review any of this, he has, you can see it has the basics, what is an iModulon, how to actually use this site. And then the crown jewel of this are the iModulon dashboards. That is what I'll step through here. Oh, maybe I'll just step through this after I finish the overview of the whole thing. And then citations. So if you want to learn more about the paper history here, there's quite a few papers that have been published and are coming out on these topics. So there's plenty of literature, even though you know, a couple of years ago there was none. And then contact info. Okay, so we'll be stepping through that in a second, but I want to just give a quick snapshot of everything. So you can see here, there's right now there's well, I think there's more than 5 data sets now because I see 6 organisms and this one has more. So maybe I'll just focus on this. So I'll show later how you can actually build an iModulon dashboard locally, but for now, let's work with what is available on the web. So there are several different organisms that all have their own ICA breakdown already uploaded. You could see B. subtilis, for example, has that microarray data set, which is what Kevin worked on initially, and I think it has an RNA-seq version now. E. coli has PRECISE1 and PRECISE2, which is an extended data set, and then a modulome version as well. Then there's tuberculosis, aeruginosa, and other organisms here that all have their ICA uploaded. So just in the interest of a snapshot, I'll go with the classic PRECISE1. This was the first ICA breakdown. And you can see the title here, organism, strain, related publication, number of samples, 280. So not even that many samples, and you can already get an excellent representation of the TRN for 163 conditions. And you have 92 iModulons associated with that. So let's just contrast that really quick with PRECISE2. And now we're from 98 to 218, or 92 to 218. So yeah, 92 to 218. So PRECISE2 is over twice the number of iModulons as PRECISE1, meaning that the data that was added to that was quite informative. Okay, so let's start exploring. So up on the first page is just a list of the different iModulons with annotation. So you can see the iModulon name, and these are mostly human annotated regulons. So there's a number that were uncharacterized here. There are some that are associated with knockouts. Here you can see here, this is what we call a genomic iModulon, because it's just an iModulon that shows up when you knock out these genes. This one too is this E14 prophage deletion, shows up as its own iModulon when that is missing. But others like CRP1 are what we call a regulatory iModulon, and these were categorized by looking at enrichment of the calculated imodulon with the known CRP regulon. And I'll show how that is done later. So you can see this is called the CRP1 imodulon, and the associated regulator is CRP. So we can sort by okay, sort isn't working for me. So we also can, there's an annotated function. So this is, again, human annotated function here. For example, this YJ has been associated with ascorbate utilization, even though it's a Y gene. You can see the number of genes in them. So RPOS is the biggest regulatory imodulon at 107 genes in it. It's quite extensive. And then there's some as small as 2. But you can see the majority of these are genomic alterations, meaning that there's probably no regulatory significance to it. It's just a genomic deletion. But there are some small regulator-associated imodulons as well. So some around 6 and 7. And then you can also see the explained variants. So RPOS is the most explanatory. FIR is quite explanatory. Flagella, CRP, these are all global regulators, so it makes sense that they explain a lot of your regulation. And then you can see precision and recall. So this is how well it actually reproduces the known regulator. So precision is if it's in here, is it in the annotated regulon? And recall is how much of the annotated regulon is in here. So the fact that this has a precision of 1 must mean that every gene, all 8 of these genes, are in the OxyR regulon, experimentally annotated regulon. But it only recovers 0.17, so 17% of that. So the OxyR regulon, let's go into this in a little bit more detail because that's the last category. It's these 8 genes here. You can see B number, the name, the gene product, and then the COG, which is the gene annotation, and any transcription factors that are associated with that. And the M sub I is the weighting in the ICA-M matrix. So you can see this is the most highly weighted gene, has to do with glutaredoxin, makes sense that that's associated with ICA. So in this case, we have that precision of 1, and you can see that a Venn diagram of these 2 is just a big circle, and so not particularly meaningful in this case. We could go to something with less overlap, maybe like that, and you can see now that we have some genes that are imodulon specific, but still quite a lot of overlap. Back to OxyR. So now we're into the nice plot. So this is what we call the ICA dashboard. It's just a beautiful representation. You have a lot of mouse overs here. You can see, so this is the actual imodulon itself. This is that histogram I was talking about, where you have most genes clustered around 0. And then you have a threshold here, and everything above this is called in the imodulon. So it's 1, 2, 3, 4, 5, 6, 7, 8. So those are our 8 genes, and that corresponds to this number 8 here. And their weightings here, 0.3, 0.3, so on and so forth. So these values are represented here. You also have very nicely the regulon plotted on here, and you can see these other regulated genes. You know, maybe there's some other things at the threshold here that might be considered here and just in this regulon, but are just a little bit lower. There's quite a few genes that don't show up in this at all, and so you know maybe they're not regulated by this signal. You could also look at it here and mouse over these genes here. It's the same type of plot. But in this case, organized by where they are in the genome. So coli is 4.6 megabases, and you can see where these are. This is often helpful if you want to see if you have a continuous regulon, right? All next to each other in the genome, all regulated by something. That's often helpful. Like you can see here, there's something interesting going on there with the sulfur regulation system that's a little bit below the threshold, but maybe it's some regulator, some operon that gets activated. Okay, so that's the imodulon itself. Let's look at its activity across PRECISE, because this is also extremely informative. So, so this is the ICA A matrix now. So this is the M matrix or S matrix. Now this is the A matrix that we're looking at. And here we can see all the different conditions in precise annotated. So these are, every column is a different sample. And so we have the baseline control, which everything is zeroed to, so it's around 0. Then you have these FIR conditions, you have acid, oxidative stress, different knock-ins, different evolutions, so on and so forth. So every one of these is a different study. And you can see if you want to, you can even click on them to get to the paper, which is kind of incredible. Can you click on these too? Yes, you can click on the individual genes too. So yeah, maybe before I move on, let me just mention that you can click on the individual genes to figure out whether it's in any other imodulons and figure out what its actual gene expression is across these same conditions. So you can do a gene by gene analysis, which is really nice. You have some options on these plots that you can play around with. So if you want to filter this, but maybe just the biologically interesting finding here is that you have these oxidative stress conditions here, deletion of SOXR, which is another oxidative stress regulon. And the result of that is that, and then this is a supplement with paraquat, which induces oxidative stress. The result of that is if you deleted a regulator and added paraquat, and you got a huge upregulation of this oxidative stress response regulon, right? So you've got this nice biological story here, where most of the time this thing is not active at all, but all of a sudden you treat it with oxidative stress and it lights up, and you know it had this oxidative stress intuition. So if you just want to understand what your imodulons are telling you about your conditions, or even what is actually relevant in your particular condition, these kinds of plots are extremely helpful. And you kind of have these mysteries too that you can dive into, which is, for example, why are these particular pseudogene repair studies showing up so high in OxyR? Maybe that's interesting to you. Also shown, the last plot, I already mentioned the overlap with the regulon, but also shown here is how, if it's associated with a regulator. So in this case, this is associated with OxyR. It shows the activity of this imodulon versus the expression of OxyR. So for the majority, you kind of have this point cloud where nothing's happening. But interesting, on the high end of OxyR expression, you see this linear relationship with OxyR activity. So that's kind of that expectation that you have a higher volume on OxyR, and you actually end up with more imodulon activity as a result. So it's really connecting these dots between the gene expression, the transcription factor activity, and all the way down to the transcription factor level. Okay, so that's what's here. You can just then, just to demonstrate some other things, there's a search page, so you can search for different things. There is various downloads if you want any of this data itself. So if you want, precise or any of the ICA for a particular organism, you can download that here. You can download all the annotation experimental conditions. So it's not just a web browser, you can get all that information. And then just going back to the About page, let me see if there's anything I missed here. Yeah, so that's the same tutorial that we stepped through. So you can see there's mouse overs on these different things that describe the different features of this. Okay, I think that's probably all we want to cover here. I don't know, Kevin, do you have any other thoughts on topics to cover here? Did I miss anything?
No, you did great. Yeah, that's awesome. Yeah, sorry about not updating those database size things. I'll do that.
Yeah, it's really embarrassing. I'm just kidding. No, this is awesome. This website is really, really professional, really beautiful, and really nice to work with. So that's great.
Thanks. It's nice to hear someone else talk about it. I sometimes forget about all these features. So yeah.
Okay. All right, so how are we feeling? Should we do the second exercise, or should we take maybe a 5-minute break first? Let's I'll have a poll set up. Okay, what I would suggest is we take one break now and then power through the second exercise and lecture, and then another break before the second exercise, if that's okay with everyone. Okay, so the time here is 8:16. If we can come back at 8:20, we can get started. Just, wants to get water, go to the bathroom. All right. See you in a minute. All right, welcome back. So for the second part of the exercise, I wanted to introduce you to the steps to actually calculate ICA for new data sets. So this has been a big initiative in our lab to gather all of the available RNA-seq for any organism that seems to have enough, or any microbe at least. Focusing especially, of course, on biotechnology-related and infectious diseases. Gather their expression data and then calculate ICA on it. And that's what we're calling the Modulome project. And there is now this very nice workflow for doing that in a standardized way. And so that's this Modulome workflow GitHub repository. If you haven't cloned it, go ahead and do that. It's really nice. It requires this other package called PyModulome, so you can just pip install that as well. What I would say as far as setup and installation, well, is that it says to begin install Docker in Nextflow. I would really encourage this if you're going to go through all 5 steps, and starting from gathering and downloading RNA-seq data, processing that RNA-seq data, and then calculating ICA. If you're not going through those first 2 steps, it should be fine to just clone the repository and run it locally without Docker and Nextflow. At least for me, that's been fine. So this might be overkill as far as installation if you're just trying to learn about these tools. But certainly if you want to apply it to a new organism, you're going to want to follow all of this so that you can do this. So what does this workflow have in it? So it's 5 steps. The 4th step is actually ICA. So the fast ICA, the clustering, everything I mentioned like that, that's just step 4. The first step, though, is to gather RNA-seq data from a public data set. You can see here, this is just a Docker run command, and it's running the shell script and calling this Python file. So it's not like an IPython notebook that we can, kind of look at, but you end up just getting all of the data available for a particular organism. I think you use certain metadata information. So you need to provide certain information on what you actually want to grab. And once you do that, this will just download it. So you can see here, it's downloading all available Bacillus RNA-seq data. Okay, so that's one thing you can try that. If that works for you, great. If not, you can use some of the E. coli examples that I'll show in the later code, or any other organism. For processing the RNA-seq data, this is where Nextflow comes into play. This is a bioinformatic pipeline for analyzing raw gene expression data. So it actually processes the reads, maps them. You have to put in all this data on, whether it's what type of data did you actually get. So you have to prepare a metadata script. I noticed this link is broken. Sorry about that. But you have to prepare a metadata file in order to download that. And then, it'll download your FASTA files and then actually process them. So that often will require some kind of high performance computing solution. I don't know if you want to be running this locally, it's up to you. You'll get this error, process requirement exceeds available CPUs, or process requirement exceeds available memory. So that can be a bad thing and might suggest that you need to run this on a server instead. We've used AWS in the past, so if that's an option for you, then you might want to look at something similar. If you have a local server, you can probably set that up as well. But those are the first two steps, is to download the data and process it. Now, step 3 then is the quality control. And this is where we actually have some IPython notebooks that we can step through. So there's 5 different checks for quality that are run. So let's just go into this. So if you clone the Modulum repository, you can go into this workflow, and then you can go to quality control, and you can open up these notebooks. So IPython notebook part 1 and part 2. And you can see that there are different, so you load your expression data and you check different QC statistics associated with your expression data. And then in the part 2, you remove samples, any samples that have poor metadata, and you check your correlations between replicates. So a variety of quality controls, and we can just step through these. So everything should here execute. In this case, we're utilizing the Bacillus Modulum data set. So that's something to keep in mind here. If you want to use the E. coli one, you'll probably have to update all of this. Or if you have another organism of interest, you're going to have to update all of that. So here you have your inputs, your log TPM. So this is your RNA-seq data. So transcripts per million is a normalized form of RNA-seq data, and that's the form of RNA-seq that we put into ICA. And that's normalized so that the total amount of RNA in the sample isn't creating any kind of bias. Then you also have additional metadata and this multi-QC stats file as well that you can take a look at these. What these data files look like in the repository, if you have that downloaded. If there's interest in that, I could click through that too. Just put it in the chat if you actually want to step through those things. So that's in data and then raw data, and you'll see there the multi-QC stats that have a bunch of this curation associated with sequencing, whether reads were aligned, so on and so forth. Okay, so continuing on, we got our, in this case, a gene ID for Bacillus and a variety of conditions. So you have number of genes, number of samples here. And you can just see these are the TPM values. They're log 2. So that's something to keep in mind here. And now you can start to load the QC data. And that's what we're seeing here. If you open that file, it's a tab separated file that looks the same as this. And you can see a number of different quality control type data sets here. And you can also load the metadata associated with all of your samples. What type of sequencing was it? Where was it sequenced? Any kind of I don't know if read depth is on here. You have an SRA ID, so you can go and look it up in NCBI if you want. A continuous version of the PMID, and so on and so forth. And then you can start to check out the quality there. So let's see, how does it actually do? So sets your tick labels pass, warn, and fail. So anything in blue is good, and then you have some things that fail. So the sequence duplication levels quite high there, and then per-base sequence content also failed that. Other things were fine. So different samples pass and fail different metrics, and you can start to analyze that and then filter by different categories. So if the sample doesn't pass any of these 4 categories, so per base sequence quality, per sequence quality score, per base N content, and adapter content. If it doesn't pass all of those, you kick it out. So that's how you end up with that high precision data set that I was talking about. The reason PRECISE works so well is because it's high quality. If you're just downloading a bunch of data and throwing it into a matrix, it's probably not going to work that well. So this criteria here are pretty stringent. And now you can see here, pass, pass, fail, pass, and it'll kick out things. And then you can mark anything that passed these metrics. You can now see how many reads you have mapped, a number of reads mapped to the different coding sequences. You can find any kind of samples that have just generally poor read depth. And then you can start to look at global correlations. I think this, here we go, global clustering, you can even cluster your data. Kind of interesting here. It actually works pretty fast. And you can see if there's any kind of weird biases or things going on in your data with clustering. But if it passes, then you can mark any samples that pass. So in this case, there's this variable pass global clustering that is here, everything that didn't get removed. So that first cluster here got removed. No, here, that first cluster here got removed. You can see it kind of doesn't look very healthy. So that got flagged for removal, everything else stayed, and that's fine. So now in the new data set, we don't have that. And yeah, you remove failed samples, and yeah, we're at 45% failed, 55% passed. So I don't know if that's good or bad, but getting things that pass these sequencing metrics is encouraging. So now you can take a look at any kind of metadata associated with your remaining data set. So now you've filtered by these criteria, and you can see what kind of metadata you actually have, and you want to make sure you curate this to the extent possible. So whatever data is good enough sequencing quality, you want to make sure that you're annotating all of this type of data so that all the downstream analyses are actually meaningful. So if something is lighting up as RPOH or some kind of folding stress, maybe it was measured at high temperature or something like that. Okay, so that's part one. Then part two, we can continue on. Hopefully all this executes as I'm doing all this live. Now we're loading all of that for these samples that we're working. Anything that didn't have good enough metadata now, you can also filter that. So that last process here, filling out all your metadata associated with your sample. If that didn't pass, you can kick those out. And now you're really down to just the cream of the crop as far as data. You can remove samples with only a single replicate, because then you don't know if it's believable or not. And then you can even check the correlations between your replicates and see if it's high enough. So I mentioned that PRECISE has a correlation between replicates of 0.98. Here we're using 0.95, so not quite as high, but still quite high. And you can see here the replicates are actually quite high for this data set, 0.98 here, so that's quite nice to see. So in this case worked pretty well. And then you can normalize this. So you always have to pick, I mentioned in the imodulome data set, if we go back and look at the activity matrices here, we can look at basically any gene, and you'll see that the control is on the far left here. It's wild type glucose. So you have to normalize everything to that one condition always. So find a condition to call your reference, and then normalize everything relative to that. So that particular condition will be considered 0, and everything will be differential with respect to that. And that's actually what you're going to break down with ICA. Now that's it. You filtered your data by these criteria. Was the sequencing good enough? Were there any weird clusters that looked non-realistic? Was your metadata good enough? And then were there replicates? So once you filter all that out, you end up with your data set and you're ready for ICA. So this is generating your own PRECISE for a new data set. Any questions on this? Okay, so I can move on then to step 4. So that was, that was step 3, which is inspect data to identify high quality data. So now we have our precise data. We downloaded it, processed it, and curated it. So now we actually compute our iModulons. So what goes into this? So this is where we actually run ICA like 100 times or something. And this is, again, not a something you really want to run in an IPython notebook. You probably put this on a server also. So default of random restarts is 100. As I mentioned before, you can use 8 cores. It's parallelizable. That's fine. And out of this, you now get your M and your A matrix. So we can step through this a little bit. So the random restart ICA, you can take a look a little bit at the Python code here. I guess the key step is when it's actually running the fast, here's the fast ICA call here. So this is that call I was talking about. You have whiten equals true. So you're now whitening your data with PCA beforehand to remove that those correlations. A massive number of maximum iterations. I assume it doesn't actually hit that. You can use a tolerance for zeros, and then the number of components. So that's about it. Under the hood, it's just running that fast ICA call that I mentioned before. You can see it's from that sklearn package here, sklearn.decomposition. It imports PCA for whitening and then ICA. PCA might actually be for dimensionality. Okay, and that's really all there is to it. It's just running that line of code on that nice matrix that you had. So if you can run this run ICA shell, you could probably run this locally if you don't mind. And then this will also run, if you want to take a look at what this actually does, this should also run the clustering. So it'll run the random restart pi. It'll compute the distance between the different runs. So it'll find the pairwise distances of all the ICA vectors to each other, and then it'll cluster it. So cluster components, and so we can take a look at that. And here you can see we're just using that sklearn.cluster dbscan. So now we're using a parallel form of it, and just clustering the components with dbscan to figure out what, with that density-based metric, what is in an iModulon and what is just noise. You know, just it only showed up in 1 or 2 of the iterations, and it's not stable, so you don't keep it. So that dbscan function call will give you that, and you can see dbscan has some parameters as well. Has an epsilon that has to do with the density metrics and how far it looks for things. Number of samples, and then in a cluster, and then this metric pre-computed. And that's it. That's calculating ICA. So the devil is in setting up your data set and getting a good breakdown. There's also a good amount of decision making in how many components you select, which isn't something that I really should probably go into today. And then that doesn't work either. And once you have your ICA, you can just characterize it in PyModulon. So that's going to be the second exercise. I'll be going into the PyModulon package and everything that you can do with that. Okay, so I think that's it for this part of the tutorial. If there are any other questions, let me know. Otherwise, I'll just get back to the second part of the lecture. Okay, so for the second part of the, is there a reason that PCA is used for whitening instead of fast ICA built in? Oh yeah, fast ICA probably does it. My guess is that the PCA step was for a dimensionality assessment. I can verify that though. The ICA does that actually. So my guess is that we find where PCA is actually used here. Yeah, it just uses the, first they just filter out the noise from the data set. It probably helps the resulting breakdown if you do a bit of noise filtration. So I mentioned before that PCA can be used for dimensionality reduction because it sorts by variance. So if you get your principal components and you sort it by variance, your very low component variance, very low variance components are pretty reliably just noise, like they're meaningless. And so to reduce noise, what is often done is just first doing PCA and then zeroing out those vectors and then recomputing your matrices. So you've effectively deleted the noise out of your data. It's not a perfect method, but I could see it helping because then ICA is going to try to interpret that noise as meaningful components, which it might try to do otherwise. Yeah. So that seems to be what the PCA is doing here. It doesn't seem to be for whitening. The whitening, I think, happens internally to fast ICA with this whiten equals true parameter. I imagine you could do whiten equals false and then do it yourself beforehand, but there's not really a reason to. Okay, so that is it for the modulome then. Let's get back to it. So far we've talked a lot about how to compute ICA, but not so much about what it's telling us. We've alluded to a little bit with some of these biological examples where it looks like a regulon shows up just by the name alone in the S matrix or the M matrix. But I really want to sell the case for how much we can actually learn with this type of analysis, because it's pretty impressive how many different types of findings you can get with this, and how many different kinds of processes that this type of analysis can capture. That really is kind of leaps and bounds above what the status quo was for us prior to this method for analyzing gene expression data. So let's get a little bit into the intuition for what is in some of these imodulons. So I've said before, they're in regulons, right? They're similar to pathway analysis, but what does that actually mean? Well, the CRA imodulon, which is also, this regulator is also called FRU-R for fructose response, or fructose repressor, is a global regulator in central metabolism, and it's pictured here from PRECISE-1. This is a PRECISE-1 analysis. I don't know how much of this has changed in PRECISE-2. But from looking at a pathway map, you can very clearly see that there is this coordinated response here along this pathway from fructose, around fructose utilization. PPSA maybe being an exception where so this is coming up, it's gluconeogenic. It might not be clear why that's involved at all. But you have these other paths away from this pathway in an opposite direction as the core pathway for fructose utilization. And it all shows up in an imodulon. And if you look just a little bit of why this is meaningful. So one, you can see topologically there's an association there. So that's great. Two, it's known that CRA is a regulator of these genes, or several of these genes, FRU-K included, and FRU-BA included. So it's already annotated as a regulator thing. So that's already validation. We can take a look at our imodulon here to just see what this actually looks like. And quantitatively, you can see these FRU-A, FRU-K here are very extensive, and then other exceptionally beyond the threshold. And other genes are maybe closer to the threshold, but they include these glycolytic genes, PGK, FBA, all along this pathway. But if that wasn't enough to convince you that this is a meaningful dimensional breakdown of your gene expression data. Again, this was extracted out without any knowledge, like no bias at all, right? This is an unbiased breakdown of the gene expression by which dimensions move independently of each other. So if the topological continuity within the pathway wasn't enough to convince you, and the presence of CRA as a known regulator of this gene wasn't enough, you can even look at the regulon activity of this on different substrates. And you can see glucose, it's negligible, pyruvate, it's negligible, fructose, it gets this big jump. And again, as I mentioned, CRA's other name is FruArt. It's always been known as a fructose responsive regulator, and you can see very clearly it just comes out of the A matrix. So this is an extremely supportive story that the ICA breakdown is biologically meaningful and can extract the real regulons, right? The functional regulons out of just a high quality gene expression data set. So let's get a little bit more into how this is done and as a prelude to broader iModulon analysis. So we've already been looking at these kinds of dashboards in iModulon DB. So maybe that was a little bit of a spoiler here. But in this case, this is an early breakdown of PRECISE. So we only had 71 independent components at this point, 4,000 genes still. But in the S matrix, also called the M matrix, the values are coefficients of the genes for any independent component, right? Any iModulon. So the values here are the coefficients, and we assign a threshold. Most are near zero. We assign a threshold and say anything above it is in the regulon. And that's borne out by presence of binding sites. The selection of that threshold has some justification. But there's always some edge cases that you're wondering if they should be in there or not. And that's probably something that can be continually curated over time. There's no meaningful direction to these. You have to assign it. You don't know, just because these coefficients are positive doesn't mean the regulation is positive. You have to consider all the signs of everything, and then you can curate it in a way that makes the most biological sense. And as I mentioned, the criteria of ICA are that the columns of S are statistically independent. So A can be dependent, but S has to be statistically independent, which basically means that you're not going to have a lot of overlap in the gene sets between iModulons. So this set of genes is not going to be appearing in any other iModulon, or not to any great extent. That's the criteria that's being optimized here, is the independence of these gene sets from each other. So they're going to be, so if you have an overlapping regulon, where you have like different modes or genes that are showing up in different iModulons, or sorry, in different regulons, they'll probably show up as different iModulons. Or sorry, as the same, they could show up as the same iModulon because they could be lumped together. Yeah. Just a comment on that curation. So once you identify what everything beyond the threshold that you want to call in your iModulon, how do you associate this with the regulator? So we do this through this process I've been alluding to, which is comparing it to previously identified regulons. So this is done through mostly through chip experiments, where you're measuring binding sites as well as gene expression activity, and oftentimes knocking out that regulator to making sure that it affects the gene expression. And this is a very slow process, but by this point, for most of the major regulators, we have some chip data out there. There's always a lot of question marks about edge cases and whether these binding sites are functional or not. And that's kind of what ICA gives you. But then you can calculate statistics on this. So recall and precision, as iModulon DB was showing. So precision says how much of the iModulon is in the regulon, and recall is how much of the regulon is in the iModulon. And so those two together kind of give you the total overlap between these two. But that's how these iModulons are annotated. So if you have a new set of iModulons, you don't know what to call these genes, your best bet is to first port over any existing descriptions and then run enrichment analysis. And PyModulon can do that, and I'll show how that works. So you can identify overlap, and here you can see this, METJ is quite a bit overlapping, and you can call this the METJ iModulon. Same with SysB, but in this case, you have one SysB regulon, but then you have two different non-overlapping SysB iModulons. And that's this process I was describing, where because they have these this requirement of independence, they're not going to overlap with each other very much. But you could have a large regulon breaking down as separate components. So this is maybe future work for ICAs to figure out what all these cases actually mean. We've started to do some of that, as I'll describe. So I showed the CRA case already, but it was really inspiring. Anand put this plot together when he was first developing the iModulon analysis. But it's really amazing how much of the core metabolic network and amino acid pathways are covered by iModulons. Basically everything outside of TCA cycle shows up in an iModulon as a continuous, topologically continuous set of regulated genes. So this is really pathway regulation right before our eyes, and it's all calculated without any kind of bias. Like the ICA did not know that there was any biological meaning to this. It was just looking for statistically independent directions out of the expression data set. So pretty cool that this works so well. I already mentioned the CRA case, so I don't want to go into too much of that. But another way, so we talked about validation in terms of topology, but we've also talked about in the CRA case validation in terms of activity, comparing glucose to fructose, because CRA is known to respond to fructose. So we've validated this more broadly by looking at these genes that are known to respond to particular metabolites. So these are the effectors in the left column, and then the expected regulator based on literature evidence. So you can see methionine is known to be a regulator of METJ, or to be sensed by METJ. And so the expectation is if we are supplementing with methionine, we should see a change in the METJ iModulon. That's what we see here in the green points here. It's the validation condition in duplicate. As if you take a look at the legend, you can see that it got repressed in response to methionine, well outside of the rest of the data set, the rest of the conditions that are available. And this was the case over and over and over, other than arginine, which is kind of interesting. I'm actually not sure why that's the case. Maybe RGR is not actually responding to arginine. It's a de novo synthesis thing. I don't know. I don't know enough about that regulon. So, but for the vast majority of the cases, if you supplement with these, then with these metabolites, then the expected regulator gets switched either on or off as expected. So this is just further evidence that these top-down kind of iModulons are actually meaningful. So far we've been talking almost exclusively about what we call regulatory iModulons. iModulons that have an enrichment with a regulator, with a known regulon. But that's not all the iModulons. I mentioned that there's 300 transcription factors. There's 150 that have been characterized, but our current number of precise iModulons is at 212, and certainly not every single one has a separate transcription factor. That would be pretty miraculous. There are other iModulons that aren't due to a regulator, and so it's really important to be aware of that and understand why these are showing up. So there's a number of different processes that can also cause an iModulon to show up, and they might even be considered to be, well, there's a lot of investigation to be done around them. First, let's just get out there. There are uncharacterized iModulons, meaning we're not sure why it's there. It's a small fraction, but it's there. A much larger fraction are called genomic iModulons, which are iModulons due to a genomic alteration, meaning something like a knockout difference, or if you're combining data that came from entirely different strains, there can be a massive expression shift that is all encompassed within a single iModulon, as I'll demonstrate. So it really kind of is food for thought. Why, when you knock out a gene, does it cause a particular transcription response? So the trivial response is that you don't have that gene there. So you're going to have a single imodulin that is that gene alone. But in some of these cases, it's more than just that. And so there is some analysis to do around genomic imodulins and understand why they're there. There's also a category which is not associated with a genomic alteration, not associated with a known regulator, but it appears that there is a functional association between the genes. So, maybe some kind of a set of stress-related genes that don't enrich for anything might show up as a biologically enriched imodulin, and that's again further analysis. So I would say maybe in the grand scheme of things, we might even want to somehow subtract these other imodulins out of our original data set, like normalize for these factors, so that we only have regulators contributing. That might be the most intuitive end goal, but that's just food for thought, not really a coherent plan at this point. But it is important to be aware that there's multiple different types of regulators, and they all kind of have their own interpretation. Yeah, so that's just showing this again, and as well as so this is the original precise data set, 94. Every slide I have has a different number of imodulins because of those little judgment calls that were made along the way. I think that precise originally finally settled on 92, but we're always deciding what the best cutoff is for some of these edge cases. Okay, so that's what's in the S matrix. It's the structure, it's topological, it's meaningful. Let's talk more about the A matrix, because there's even more to be learned there than just you supplement something and it affects the regulator. It's great that it does that, but it's much more complex and interesting than just that. So when we're looking at the A matrix, now we're not saying anything is uncorrelated anymore. All of the statistical independence was enforced in the S matrix. So these can now be correlated. So imodulin activities can be correlated with each other, and there's a lot to learn there. And that's one of the topics we'll talk about. But we've already talked about the dashboard, the A matrix dashboard. You have an imodulin activity on the y-axis. On the x-axis, you have all your conditions. And depending on what the imodulin is and what the activities are and patterns in the activities, you can learn a lot about your activities, what that imodulin is actually doing. For example, you might have some condition that you really don't understand what's going on within the cell. Let's just say as an example, your cells aren't growing very well and you don't know why. Well, if you measure their RNA-seq, you might find that certain stress imodulins that lit up in other conditions, like oxidative stress, are also lighting up in your condition. And so you could diagnose, you could say, oh, my cells seem to be experiencing oxidative stress because they're experiencing the same patterns as an oxidative stress control study, right? So you can learn a lot from this kind of comparative analysis of activities. So it's really, really extremely powerful to build up this data set, and more importantly, have very good annotation of your conditions. And that's why the metadata step in the modulome was so important. So let's go into a couple of the types of relationships that can exist between imodulins in the A matrix. So maybe the most simple and expected is if we have a regulator expression level, which we do, we might expect that if we have an imodulin for that regulator, that its activity would be correlated with the expression. In the case of RPOS, you could see it is, and that's very encouraging. It's not required, but it is great to see that in the data. So as you get more RPOS, it's known that RPOS regulation is RPOS dependent. RPOS is a sigma factor that actually has the exact same -10 box motif for binding DNA as sigma 70, the main one. So normally they would compete. So to actually regulate RPOS activity, it has to regulate its expression, it has to change the availability of this transcription factor itself. And so that's what actually controls its activity. So for this regulator in particular, you really would expect this correlation to show up, and it's great that it does. In some cases, you get this kind of interesting plot, which is kind of like a switch-like response. So below a certain threshold value, really doesn't matter what the expression level of this thing is. There's not enough of it to activate its regulon for whatever reason. Maybe it's competing with some other things that are also bound to those same promoters. I don't know. I don't know how to what extent we've looked in this particular case. But after that point, you see a linear response. So now you have this kind of switch-like into a linear response kind of activity. So for some regulators, it looks like that, which is really interesting. In some cases, you have these outliers, which I guess you would have to look into their specific cases to understand why they don't fall on the general trend. Could be some kind of nonlinear effect. It could be a very different genotype or something, mutation that showed up in these that causes them to have a very different behavior. That gets into another one of the assumptions of ICA, which is that there's genetic identity of the DNA so that they're all affecting the same sequences. And that's another can of worms that's probably not worth going into at this point. And then last, you could, beyond just expression of the regulators with the imodulons, you can actually correlate imodulons with phenotypic effects like the growth rate, phenotypic factors. And here you can see we have a imodulon associated with translation, so containing things like the ribosome. And we know that ribosome amount correlates with growth rate. So we might expect that expression of the regulator of that, via the translation imodulon activity would also correlate with growth rate. And indeed, we do see a correlation there. And this association underlies one of the more interesting and predominant trade-offs called the fear-greed trade-off that I'll discuss in a bit. So you can also do these kinds of phenotypic correlations, and those are meaningful as well. Okay, so as I mentioned, you can learn a lot by comparing the activities across conditions. So this is this B subtlest case that Kevin probably generated. And you can see, you can watch this FADR, this fatty acid utilization imodulon over time because they did a time series of RNA-seq. And you can see prior to glucose exhaustion, there's almost no activity of this. And then a big upkick as it tries to scrounge for other carbon sources to maintain its energy. And then I guess it adapts to that state eventually. And then you have also an uptick in stationary phase, which is kind of interesting. Similarly, you have this Lexay DNA damage response that gets kicked up under H2O conditions, H2O2 conditions. But interestingly, also during some population kinetics around swarming, which is an interesting association that you might not expect and could, suggest some links, right? Like I said, maybe your cells are experiencing an oxidative stress or DNA damage during this behavior for some reason, right? So you can learn, or you can come up with new hypotheses based on that kind of comparison. The other visualization that is quite often used is what we call a DEMA plot, or differential imodulon activities. Now here, we're not looking at it imodulon by imodulon, we're looking at it condition A versus condition B, which imodulons are most different across these conditions. And that should tell you, what are the core regulatory differences that the cell is sensing, right? Like what does it think is different about one condition versus another? Has it gotten additionally stressed in a particular way, right? So rather than comparing thousands of genes in a differential gene expression plot or a volcano plot, now you have a much reduced set of variables. And if you filter this by a threshold, now you're just looking at 5 or 6 or 10 or so highly interpretable variables. So this has been an extremely useful plot for us. And you can generate case studies like the one below that are maybe not that critical to go into. But just to give more intuition for why a DEMA plot or an imodulon type differential expression analysis is better than classical differential expression analysis, or can be better, this is a really good intuitive case. And this is also an argument for any kind of pathway analysis or any kind of gene clustering. But this is a particularly powerful way of doing that breakdown. And that's what we're seeing here. So we're looking here at RPOS imodulon versus translation imodulon. So this is a fear-greed trade-off that we'll talk about, where you have a growth-related thing and a stress-related thing, and they are anti-correlated. So the more stressed you are, the less you grow. So we see that over and over. And you can see here that there are these RPOC mutations that kind of restore your cell from a more stressed condition to a higher growth, less stressed condition. And if you look at a DEMA plot then, or sorry, a differential expression plot of RPOB expression versus a mutant of RPOB expression, what's really interesting is what's highlighted here are those 100 genes in the RPOS imodulon. And you could see that there's a almost uniform decrease in their expression, but almost at a differential gene expression level, almost none of them pass the cutoff individually. You only see it if you group them by their shared presence in the RPOS imodulon. So therefore, looking at the imodulons should be a more meaningful way of finding these coordinated changes. It's a smaller change on an individual level, but certainly once you group 100 genes that are all changing at this level, it's a massive gene expression change. So this is just an argument for why these kinds of imodulon analyses are more powerful in many cases than the classical differential gene expression analysis. Just a little bit more on this type of correlations that can show up. Some more cases here that are kind of going towards Emery's question about what does it mean that you have these independent or these regulons that share as few genes as possible. Well, oftentimes what you end up getting is different regulators, different imodulons for the same regulator. But they'll show correlations in the activity matrix instead, or mutual information in the activity matrix instead of the structural matrix. So here you can see CRP1 and CRP2, and you have this interesting kind of Michaelis-Menten curve here. And another interesting point here is that the delta CRP, so the knockout of CRP, ends up at this really far extreme that has almost no activity of this one. What's really interesting, I don't have a full expose here. There are two binding modes of CRP. There's two ways that CRP can bind, type 1 and type 2. And if type 1 is, if one of those types is tighter binding than the other, you can actually end up with this type of curve. So we actually think currently, I believe, that this is showing up in two different imodulons and with this relationship, because it's these two activities of CRP, basically low CRP activity and high CRP activity are showing up as different imodulons. Yeah, the result of that is this coordination between these two. You can also see classic responses. ARC-AF and R are known to be associated with anaerobic metabolism. You can see them showing up with this anti-correlation here. Anaerobic and nitrate respiration, which are these anaerobic conditions, showing up at the extremes, whereas all these aerobic conditions show up at the other end of the spectrum. So that's a very expected relationship. And FER2 and FER1 also have a similar thing to CRP1 and 2. I'm not aware of them having different binding conditions, but you can see the delta FER knockout is up here instead. And there's also some iron conditions that end up on the other end here. So analyzing the properties of the A matrix can be very informative for understanding the coordination of regulators, which is beyond really what ICA is supposed to be able to do. It's supposed to be able to find independent components, but here you can see correlations between these components. And this can actually be formalized through clustering. So now we're talking about DIMCA plots, or differential imodulon cluster analysis, where we can find clusters of imodulons that that have related activities to each other. So they tend to move together. You could see RPOS here is clustering of pro-VWX and GAD-EWX. So this is a stress response, and these are acid stress responses. So it makes sense that these activities would be coordinated. And now what we're talking about is beyond the regulon. We're talking about the concept of a stimulon, or multiple regulons that act, that all respond to a single stimulus, in this case, some kind of stress response. So we can really get a very deep understanding of the structure of regulation by this higher order analysis of your ICA matrices. And you can see there's quite a few meaningful clusters here. This cluster 10 is the biggest one. And I'm not sure if we have a full interpretation there, but you can see CRP1, 3 different CRP imodulons are within that cluster. And so it's almost certainly some kind of global energy regulator or alternative carbon regulator. Maybe MALT is there, another carbon source, something like that. So that's very interesting. And now I just have, for the rest of the time here, let me see how many more slides I have. I just have another set of case studies that, I mean, I don't really have to go into all of them. I could probably just go ahead to the imodulon analysis, but these are here. If you want to go into any of them, they're in the slide deck. And it's really just more of this the same story. You can learn so much from looking at your ICA S matrix, comparing it to regulons, and comparing it to ChIP-seq, ChIP-exo data, the actual binding sites, mapping them onto pathway maps. And that tells you that these are meaningful structurally. And you can look at the ICA A matrix and tell a lot about how your cells are regulating themselves and the coordination across different regulons in some shared stimulus. So yeah, I think I'll probably close off there. And then for the last activity, I'll just step through the PyModulon package, which is for ICA analysis. So once you've calculated your imodulons, how do you actually analyze them? What can you do with them? So I'll take any questions before I move on.
Maybe just one quick question. I'm pretty sure I've asked this before, but you said you can characterize components so that the activities translate to expression. So in the previous example, I think slide 46. Yeah, can I assume that activities mean higher expression in one modulon in here?
Yeah, so this is literally the ICA A matrix value. Now, if you want to go from this to the quantitative expression, you have to multiply them by their weights in the S matrix. So it's not really one-to-one, like this is increasing twofold or something here. You kind of have to analyze them relative to each other.
Okay. Yeah.
You know, everything's just on a different scale here. Okay, so let's do then a quick overview. I think this last tutorial won't take too long. So the PyModulon repository is here. You can clone this, but you really don't have to. You can just install it with Python pip install and then start to use it. There's a number of optional dependencies that I would recommend, especially if you're going to be doing your own imodulon characterization. The meme suite does the motif enrichment of imodulons. And this isn't really something I touched on a lot, but whenever you're trying to do a new annotation, or even if you have any kind of imodulon that you think is regulatory, that you think is due to a regulator binding in front of that promoter or somewhere around the promoter, you're going to want to do a motif analysis. So what you do for a motif analysis is you, and I'll show you in the code, PyModulon, but you grab the promoter regions for that and you pass them into the meme suite to identify whether or not there is a shared motif in front of these promoters that would justify that, hey, these probably have some regulator binding there. And then what the PyModulon also does is says, okay, I found this motif. Does this look like any known motif? And that's where it pulls in TomTom, which is, so the meme suite has a variety of different motif finding features, from finding them de novo, finding gap motifs. You can also take a known motif and scan for it. But what TomTom does is it compares a motif you have to a database of motifs and tells you, does this look like the motif of a known regulator? And then you could say, okay, like it overlaps the regulon, it has the same motif as the known regulator, it's definitely regulated by this. So that's a really important and useful characterization of your iModulons to find an actual binding site motif. And so I would recommend downloading this, even though it's a little bit of a pain to download and get working. But if you don't have this, you can run the rest of the code, but you won't be able to run that part of it, just so you're aware. But this is quite easy to install. I don't think there's anything particularly special about this. What is special is the documentation. So the PyModulon documentation is quite extensive. Has a little bit on the, iModulon about, a little bit on Docker. I haven't had to use Docker personally for this, but you're welcome to. And then it has a number of these tutorials on different things. And so I can step through in the short course. If you look in the short course, you'll see a PyModulon tutorial there. All I did here was just grab the first set of tutorials from this documentation, just so you'd have it in one place to make sure that you can run this, just to make it a little more point and click for you to get started. But I didn't, copy and paste the whole thing. So what I'm going to do here is step through these basic steps to understand how PyModulon stores the iModulon data and some of its capabilities, visualization capabilities, and then I'll go through the documentation on some of the more advanced capabilities. That sounds okay. So where we left off with the Modulon workflow is that we imported the available RNA-seq data, or we would have, processed it, curated that data, and then ran ICA on it. So ran it 100 times, clustered it, found the consistent components, right? So that's everything so far. So you've ended up with an ICA and an A matrix, and that's where PyModulon starts. That's this step 5 in the workflow. So if you go to the workflow here, we're now in step 5, characterize iModulons using PyModulon, okay? So we're still following the original trajectory, we just have to switch packages. So what can PyModulon do? Well, it's its own package, so you import it, and you can just import, it has ICA data and it has example data. And so these are two variables that we can use that are kind of ready-made. So I think the first thing to do is inspect what's in these, because it's quite a few fields, including metadata, so that you're aware of it. But even if you're not, we can step through some of the basics here. So in example data, it already has an ICA breakdown as well as the raw data itself. So in example data.m, this is our ICA M matrix, and we can see we have all the genes here, as well as, in this case, this is a precise 1 data set, so it's 92 iModulons. And then from the heads, you can see it's the same iModulons we've been looking at in iModulon DB. So this is now a code representation of everything in iModulon DB. Then we can look at our A matrix as well, and it's those 278 conditions, same as in iModulon DB. Now from this, we can construct, although we've already imported it, but we can construct an ICA data structure. So this is now a pandas data frame that is different than the example data file. And so this can be desirable if you want to use those types of built-in functions, right? So sometimes we'll be using example data, sometimes we'll be using ICA data, but you can extract different fields from it. So ICA data, you can grab the gene names, which are here, the sample names, the imodulon names, whatever. It's all in this ICA data structure, and you can just grab them through the attributes. Okay. But we also have our raw data here. So now we have the same genes, but this is just the raw samples. So it's the same columns as the ICA A matrix, right? But instead of the rows being imodulons, the rows are genes. So this is our raw data matrix. This is precise one, this data set. And so if we want to do anything with that, we can. Otherwise, we can just we can grab that, put that there, and we can start to just look at things. So the first thing we can do is annotate some of these things. So now we have an there's a gene table. This is one of the metadata that is comes with the example data here that you would have to construct for your organism if you want to do this. This doesn't, come from anywhere in particular. You just have to have this for your organism, and you can construct this gene table. It has where does the gene start and end, what strands is it on, its name, how long is it, what operon is it, what kind of COG annotation, and what is its accession number. Okay, and then you can do the same for samples. And we have a bunch of sample metadata as well. So our first condition is wild type glucose. This is our control. You can see our study and project is the control. That's what we normalize to. I already mentioned that that's a key step to pick a condition to normalize to. And then you have a bunch of other things, like what electron acceptor, what carbon source, and all of this, I'm sure, is helpful for follow-up meta-analysis. Then last, you can show your actual imodulon stats. So now we have the imodulon name regulator, and now this is what is on imodulon DB. You might remember we saw the precision and recall there, right? And now we have the number of genes, p-value for enrichment, I'm sure, an F1 score, which is calculated from precision and recall, and yeah, threshold. Dynamic threshold for that imodulon for calling it in that gene. I don't know if that's helpful or not, but it's there if you want it. And yeah, now we can put that same information into our ICA data variable. So we've moved it from example data to ICA data, and now we're working in pandas land, so we can use functions like num to name and grab interconvert between our B numbers and our gene locuses, loci. Okay, so that is everything that's in the basic data structure. It's very straightforward, but you can explore everything that's in ICA data, and we'll pull out, in example data, and we'll pull out some more things. So now what can we do with this? Well, as I mentioned, one of the main things we want to do is compare our ICA with the regulators. So where do we get the regulators from? Well, Regulon DB has Regulon annotations, but fortunately, the example data also has a TRN in it, and you can see it's here. It's in example data.trn, and it has these different fields that you should probably populate if you want to do this. Things like evidence are very helpful. So if you want to do this for your own organism, you're going to have to assemble this data structure. So you can see the regular FMN has regulates this gene, and it's known to be a negative regulator. So on and so forth. So there's a variety of, it's a representation of the TRN in terms of a regulator, a gene that's regulated, and what is the effect of it. And you can see, and now we're just putting it into the ICA data variable. And we can now view things like an imodulon. So for ICA, we can do call the function view imodulon and grab an imodulon name. And we can see these are the 8 genes in the GLPR imodulon. These are the weights, these are where they are, whatever. You can just see the properties of this imodulon. We can also do it at a gene by gene level, where we take a look at imodulons. If say we want to say like the first gene here, right? I'm like, okay, that gene is interesting to me. GLP-Q. I wanted to learn more about this. What imodulons is it in? And so I can use this function imodulons with and figure out what imodulons contain it. And in this case, it's only this one regulator. And you can use the same thing, but with the gene loci. And this is a different example with a different imodulon. You can also rename your imodulons. This is just kind of curation. So if you're building a new set, you can rename them and the names stick, and that's all great. Okay. So that's the basics of the TRN structure, but there's much more you can do with TRN enrichments that we'll get to in a second. But first, let's talk a little bit about visualization, because we had all these nice plots in imodulon-DB. And of course, we want to be able to generate these live dynamically. And so let's talk a little bit about how to generate those plots. So here I'm now creating a new ICA data variable that's just from load E. coli data. So I'm not building this from scratch anymore. It's just, you can take a look at this function to see how this is built, but this has now more metadata in it than the previous example. But if you just want to, so we're going to start out by just gene expression plots. So not imodulon plots, we're just looking kind of at PRECISE itself. So very simple function for this called plot expression. And you can just grab a gene and plot it for your ICA matrix, or sorry, your ICA X matrix. So now this is, again, labeled with all of our conditions, just like in imodulon-DB, and it's the expression of our gene B002. And you can do some nicer things like extract particular plot projects. You can do it by gene name instead of the locus ID. But that's about it. And then at the imodulon activity now, instead of plot expression, the function is plot activities. And you can do the same thing. And you can even highlight a particular study. So here it says highlight CRP. And you can see in nice, pretty colors your study of interest, which seems kind of like bells and whistles, but it looks nice. And you can even extract that part if you want to and not see the rest of it if it's getting a little small to see. You can also just plot the metadata to see if there's any conditions of interest, in this case, plotting all of the growth rates. And so if you want to grab any of that data, you can. All right, so those are the basic plots, and you saw the same ones on imodulon-DB here, this type of thing. No, at the gene level. So we have to go here. Yeah, this type of thing. So what about imodulons? Where do they come in? So the scatter plots are really what we want to do. Well, yeah, so we're already plotting imodulons there. But in terms of more advanced visualizations, we also have scatter plots available, and you can see all the options here for coloring, labeling, etc. With some examples, but the basic function here is just plot gene weights. And here, now we're just viewing an imodulon. So it's the same thing where the x-axis is the gene location in the genome. So it's from the origin to the terminus, and it's 4.6 megabases. So this is, this is the E. coli genome we're reviewing start to finish. And you can see what I was talking about earlier with these contiguous operon type imodulons, where all of these genes are spatially right next to each other, and here as well. So you kind of have 3 different spatial clusters. So that's good to be aware of. So you really only have probably 3 promoters that you're actually talking about being regulated here, even though you have something like 10 genes or 8 genes, whatever it is. And you can do the same thing with more complicated imodulons. And yeah, you get all your labels of the additional cog categories. Yeah, and you can label. So in terms of your optional values, you can label them with gene names. You can see how quickly that gets messy. So I wouldn't necessarily recommend doing this unless you either have a you set a larger threshold or you have a smaller I-modulon, something like that. But that should be enough to explore your conditions. So you can also compare two conditions. We're talking about DEMA plots, so you can compare expression of particular I-modulons here, FNR expression versus ARC-A expression. Sorry, this is comparing expression of particular genes, or different activities. So this is genes, and this is the activities of these. And in both this case, you can see it's kind of an outlier down there. And then you can also look at gene weights across samples and see if there's any outliers. So sorry, across I-modulons. So in this case, you have two thresholds here. We're looking at the M matrix here. So everything above this threshold is in this I-modulon. So everything above this line here is in this I-modulon. Everything to the right of this line is in this I-modulon. And you can see they're almost entirely separate. That's the expectation of ICA, but they do share this one gene SBP. And so that's their one correlation between them. But you can see if you did the correlation between these two, it'd be effectively 0. So then maybe the last basic plot to go into is the differential I-modulon activity. These DEMA plots that I was saying. So you have two conditions here. Here you set up two conditions, and it highlights the genes that are changing the most. So the conditions here are wild type with iron, and then deletion of the FIR, which is the iron uptake regulator with iron. And you can see the major I-modulons that are differential are FIR, iron-related, zinc, metals, basically. So that's very intuitive. It's a nice biological story that just immediately falls out of this kind of differential I-modulon analysis plot. And then, yeah, the last one here is just a similar example with more options. Okay, so that's everything I wanted to go into in terms of a direct tutorial. The point of this was to basically just make sure that this is running on your machine, just to give you an IPython notebook to get started with enough example code. But I want to emphasize that PyModulon can do a lot more. As I mentioned, let me go to the related slide maybe. When you can calculate activities for new data without requiring a new ICA breakdown. Let's say you generated a new condition for E. coli. You can analyze that. You can get your activities for that condition without recalculating everything. And you do that through this tutorial here, inferring I-modulon activities for new data. And you can only do it for genes that overlap with the existing ICA data set. So you can't do it for any new genes, obviously, if for some reason you have new genes in your data set. But the key function is just this infer activities. And then you get your A matrix values from that. So that's there if you want to do that. And it's just a single function call, no real issues with that. You probably want to, the other thing that I should mention is you have to run this first. I mentioned all of your RNA-seq data is normalized respective to your reference condition. So if you ever have a new data set, you have to make sure that's also normalized with respect to your reference condition. So you give it your reference, it has to be mean 0, and you take a differential with respect to that. So you have to pre-process any new data that same way. Another really important feature is this enrichment between your I-modulons and the regulons. So I showed earlier how you can get your TRNs, this TRN variable here. And very broadly, you can compute enrichment from any set of genes, your gene set, target genes, and then all genes, and I'll say how enriched was this. I'm not sure if it's using a hypergeometric test or what there, but maybe that's something I should look into. But it does some kind of enrichment calculation for whether or not these genes are have significant overlap compared to all genes. And you can do the same with a Regulon, but you can, if you have your TRN there, this TRN variable set up, this TRN table that I mentioned earlier, you can even just call it by name. So you could say, okay, here are my genes, and this is the Regulon. I want to see if it's in this Regulon or not. And you can see in this case, it actually is, has a p-value of, you know, it can reject it, has a precision of 1, so on and so forth. So this gene set is the LRP Regulon, or is a subset of that, at least. You can also, there's a wrapper to do this for the entire TRN. So you can do an enrichment against all Regulons and see for the set of genes, is there any Regulon that it gets enriched in? So that's very helpful when you're trying to just de novo annotate a new gene set, a new iModulon as like, what regulator is this? There's just a wrapper code there for doing that. Yeah, I'm just saving that. Now, once you have your iModulon, I mentioned another way to validate this is to find a motif and a motif at the promoter. And so for that, we have to use this meme and so meme suite. And so you actually have to install this, and that's something to be aware of because that's not going to work otherwise. And there's quite a few variables here to play around with. Specifically, you have to decide, okay, you think that there's a regulator binding around this promoter, but you have to decide what is the region that you want to search for it, because it's not necessarily going to be in a particular location. Sometimes they bind upstream of the core promoter element. Sometimes they bind on top of the -10 box. Sometimes they bind downstream of the transcription start site. So it's really hard to say exactly, and you might have to play around with this, but there's some defaults there. That's a pretty wide range. That is a pretty wide range. So it's probably not too bad to just use defaults if meme can give you a motif there. And yeah, you can set whether or not you want it to be palindromic. Lots of motifs are palindromic, but it's more flexible to say to not require that. And yeah, some other thresholds and cores and things like that. So then this will do that. It's just this find motifs function. And you can see all the sites here that get spat out. And what the sequences are, how much they overlap with that motif, and some other data here. So this is really nice to do when you're trying to annotate a new iModulon. Maybe you find that there's a motif in front of it. You can hypothesize that there's some regulator binding there. And then if you're like, what regulator is that? Say you find a motif, right? So now you've run it on your set of genes in your iModulon. You find a motif. That's great. What regulator is it? It's not going to tell you that initially. You have to use TomTom to do that. So you use TomTom to compare your motif against external databases, and it will tell you what the top hits are for your motif in terms of the target and some, yeah, just a sorted list of what the top are. And here you can see RHR is by far the top hit. So that's another nice feature. There's some stuff on thresholding. I mentioned before, how do you actually decide what is in your iModulon or not? And I mentioned this D'Agostino cutoff. And so there's code for doing that. And so that will help you determine for a particular iModulon, what should the cutoff be. It also has a k-means implementation if you want to try that. So this is everything that I was talking about. And then your iModulon membership will change depending on what your thresholds are. And it could be an iModulon specific threshold, or maybe even should be an iModulon specific threshold that you're using. So you can recompute the threshold and take a look at that. You can manually change it there, whatever you want to do. there's some stuff on comparing imodulons across runs and across organisms. So sometimes you're going to be running this with different data sets, or sorry, different parameters, you know, or you know, increasing data sets. I mentioned all kinds of things about, yeah, as you increase data, how do your imodulons change? And there's code for generating these types of plots showing how the imodulons relate to each other. So you can check that out. There's information on how to generate your gene table. So you can annotate your genes to run all these functions, basically. So there's some optional things like the CAG and the COG that annotate the genes I think everyone will probably want to use if they have that available. But this is just some additional help on that. Eggnog is another functional tool for annotating genes. So all of this just helps you populate your gene table with annotations to look. You could look for enrichment. You can just print it out, but it just helps you with annotating your imodulons. And then maybe last, yeah, there's some code on generating single variants, single gene imodulons. Oh yeah, the explained variants. This is maybe important. So one thing I didn't mention before was how do you calculate your explained variants? So I mentioned that imodulons don't de novo tell you how important they are to reproducing the original data set. PCA does. Your first component explains the most. ICA, you have to calculate that after the fact. And so there's this function explained variance to do that. Given ICA breakdown, how much of the original data does it actually reproduce? But then there's some additional code here. So this is how to identify imodulons that explain the largest fraction of variance for the data set. So this is all the imodulons. Sorry, this is all how much of variance is explained by all the imodulons. But you can do it one by one. And this is that result that I was showing earlier, that RPOS explains the most on its own, most variance on its own, which is expected because it's such an important global regulator. So that's a pretty intuitive kind of result, and you can plot that. That's all good. There's also, I'm not going to go into this, actually, probably call it here, but there is a tutorial on actually how to build those imodulon DB dashboards. So if you have an organism of interest and you want to go through all of these steps on your own and contribute a new organism to imodulon DB, this is where you'll start. Yeah, a new organism here, that's where you'll start. You can yeah, clone the repository, set up the necessary data frames, and you can build these dashboards. Yeah, this is a really long tutorial, so I probably won't step through all of this, but if you are interested in this, I would suggest reaching out to Kevin, who has put all this together, and he can help you out to get installed there. So that is it. I think just to summarize, the topics we covered today are how to calculate imodulons, what imodulons actually tell you about the biology or the transcription regulatory network of a organism, and then finally, you know, what kind of cool biological findings can you learn, and how do you actually use PyModulon to get there, right? How do you take an existing ICA matrix, which might be where a lot of people start from, and use that to understand their system? Maybe you have a new expression data set and you just want to run this procedure on it and compare it to all the existing data sets. Hopefully that example code that you have now and being set up with these workflows will enable you to do that. So I don't think it's worth it to go into these last case studies, but there's plenty more of cool biological stories of what imodulons have taught us. If you're interested, go through them. There's related papers published on many of these, and feel free to reach out if there's something related to your work that you want to learn more about.